Hello spectators, I am Subhradeep and on my presentation I am going to discuss on RNA polymerase. Here is the headings. As you can see, I will try to explain it properly. So let's start. So first of all, we should know what is RNA polymerase. RNA polymerase is an enzyme that synthesizes RNA from DNA template. And we know that synthesis of RNA from DNA is called transcription. That means it catalyzes the transcription process. Here I have mentioned some data. I will try to explain them. In case of prokaryotes, single RNA polymerase synthesizes all types of RNAs like mRNA, tRNA and rRNA. But in case of eukaryotic cells, for three kinds of RNAs we need different RNA polymerase. For rRNA, RNA polymerase 1 is needed, and for mRNA, RNA polymerase 2, and for tRNA, RNA polymerase 3 is being used. First, let's talk about prokaryotic RNA polymerase. It is a complex holoenzyme. So first we should know what is holoenzyme. The holoenzyme is a compound where an enzyme associates with another core enzyme. As we know what is holoenzyme, here we can see that the core enzyme forms a holoenzyme with the addition of sigma subunit. And the other informations are that each bacterial cell of E. coli has about 1000 to 2000 RNA polymerase molecules, and its molecular weight is 480000 kilodaltons. Here is a diagrammatic representation of RNA polymerase. This is the core enzyme and this is a sigma subunit. The core enzyme consists of 4 subunit, actually 5 subunit. Omega subunit doesn't have any activities as it is only for enzyme assembly. And then there the sigma factor comes and forms the RNA polymerase holoenzyme. We already know about the subunits, that is 2 alpha subunits, 1 beta and beta prime subunit assembled by omega. Here is some information about them. The alpha subunit, the beta subunit and the beta prime subunit and the omega subunit. Here the alpha, beta and beta prime subunit are coded by RPO ABC genes respectively, and the omega subunit is coded by RPO Z gene. Just like omega subunit, the alpha subunit also helps in enzyme assembly. Here the beta subunit, it is the most important as it helps in the elongation process. It binds with the incoming nucleotide to be added to RNA chain and helps in the formation of fast phosphodiester bond. The beta prime subunit is also a catalytic unit similar to beta subunit. We have already discussed about omega, that it helps in the enzyme assembly and shows no enzyme activity. Now the sigma subunit. Here is some information like it's formed by single polypeptide chain and coded by RPOD gene. It has the major role, that is the recognition of promoter region. After recognition, the core enzyme binds with DNA strand. There are some 2 sequence, -10 sequence and -35 sequence. Sigma subunit recognize them. -10 sequence is called Prevno box. Here I mentioned that after 10 nucleotides joining, this subunit remove itself from core enzyme. But there is another fact I hear about is when the elongation speed takes place 40 nucleotide per second, the sigma subunit separates from the core enzyme. Now see RNA polymerase function. Well, we have already discussed about it, but as it's mentioned here, let's see again. First point is unwinds 50 bases of DNA. What does it mean actually? RNA polymerase unwinds about 50 bases of DNA around the initiation site to form an open promoter DNA complex and provide single strand of DNA to act as template for transcription. The second point is catalyzes the formation of phosphodiester bonds. That means RNA polymerase basically catalyzes the formation of phosphodiester bonds between successive nucleotides of a polynucleotide chain during synthesis of both DNA and RNA. With sigma factor, it contributes to synthesis of RNA because sigma factor recognizes promoter site. And the last function is Enzyme RNA polymerase catalyzes the transfer of nucleoside monophosphate from nucleoside triphosphate to 3' end of growing polynucleotide chain, because this releases energy and sets free pyrophosphate group, which is needed for transcription. Now the eukaryotic RNA polymerases. I already mentioned them before. Let's see their products. RNA polymerase 1, which is found in nucleolus, synthesis all kinds of rRNAs, that is 828S rRNA, 18S rRNA and 5.8S rRNA. RNA polymerase 2 found in nucleoplasm, synthesis M and SN RNA, and at last RNA polymerase 3 found in nucleoplasm, also it synthesis tRNA and 5S rRNA. So these are all information about RNA polymerase. If any one of you have any doubt or question, you may have asked in the comment section. Thank you for watching.